This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
All right. Hey guys, this is Razib with the Unsupervised Learning podcast. So I'm here with my friend Antonio Regalado of MIT Technology Review. Those of you who have been following my oeuvre, my body of work, my activities over decades now, know that I do have a connection to Antonio. He is the journalist who broke the story about my older son being whole genome sequenced when he was in utero. The first documented. I always believed at the time, and I think I told you this, I think somebody else has done this and it will come out over the years, but nobody else has stepped forward to claim that they had their offspring sequenced in utero before they were born. Right, Antonio?
Hey, Razib. Yeah. Yes. I just want to say, I've been reading your writing for a long time, 20 years probably. So
yeah, I know. We don't need to talk about how old we are, bro.
I'm familiar with your work. It's been great. Very immersive writing about genetics and history. Yeah, good question. I did when you sequenced your son. That was an interesting story because this was sort of in the frame of sort of do-it-yourself or direct-to-consumer genetics, but kind of the extreme version. And I just had the feeling that somebody ought to be doing this, and so I reached out to somebody on the West Coast, former grad student of George Church, who was doing prenatal sequencing of some kind. And I said, do you know anybody, have you ever heard of this? And he said, well, you should talk to Razib. And so that's how the story came down. Yeah. I think at the time I did find that there had been whole genome in utero sequencing in the case in a research study in which they were looking at, medical genetic issues. So I think the way I framed the claim was yours was the first healthy person to be born with their genome sequenced. And that was interesting because what were your reasons, right? I mean, just curiosity, just for the love.
Yeah.
I mean, that was just because you could.
Yeah, that's basically what that ended up because I just felt at the time, well, we can do this, why not? And people were saying, this isn't like what I was going to, because we were talking about CRISPR, just so people know. But, people were saying, oh my God, did you get an IRB and all this stuff? And I'm just like, what I told you at the time is, look, at the stage he was at, we could have just gotten an abortion. It's like, the fetus does not have personhood. So the fact that people were acting all weird about it, to me, was very confusing at the time. And I think it was just unfamiliarity. And I think that they've done a lot of whole genome sequencing. I know in low coverage in China, in utero, right? They've used that sort of thing before. So it's not that big of a deal. It's just information. And I thought someone should do it. I did it. I had assumed other people had done it. I wasn't doing it because I wanted to get in the media. You just randomly found out about it because I had been asking around in terms of the logistics, and that's, so it was in the grapevine, or it was in the network, the Genomic Sciences Network out there. So yeah, that's how you broke that story. And I think run into you at conferences before, your genetic conferences, and you reported on our field on and off for decades now. And what I want to talk to you about today, so I did a podcast with you, and I'll probably post a link, actually. And remember to post a link for the Insight. My previous podcast with Spencer Wells, we had a podcast where we talked to you about your, a profile of He Jiankui, but basically you covered the CRISPR gene editing, CRISPR baby story. I think it was 2018. Was it 2018?
It was November 26th, 2018.
Yeah, okay. So you covered that. You were pretty, early on that. And We just talked about what the implications would be. It's 2024 now. He has come back on social media. Also, CRISPR is, it was already mainstream, and just for the listener out there that's not conversant in the field or in the genetics, CRISPR's been around in bacteriology since the late 1980s, and then basically they realized around 2012 that they could use CRISPR-Cas9, which is basically it's like a bacterial defense against viruses, I believe, is what it was originally evolved, quote unquote, for. But basically, you can like have a really, really good, accurate cut out of the genome. And then, you can obviously spice stuff back into the genome. But the key is it's extremely cheap, extremely easy, and extremely specific. And so the whole idea was this would democratize gene editing, which apparently is a term I read in your piece that only came about in 2005. I was surprised by that. But genetic engineering, right? Genetic engineering for the masses. Before CRISPR, genetic engineering was a very, very niche affair. Companies like agricultural companies would use genetic engineering because they could create, I don't know, like some squash that like was, immune to frost. Okay, well, there's huge, huge economic upsides to that. So they can invest in a laboratory that did like the recombinant DNA technology and all this stuff. And, it was a very, very laborious process, required a lot of input. CRISPR, opened up the possibility that people could do a lot of different things. But the other aspect of it is people are really scared about what this might entail. And, that obviously came up with the gene editing of the two babies that He did in 2018. Your piece is called Unnatural Selection, which I think gets at the idea that once this becomes ubiquitous, it's going to be kind of a new force in evolution. Now, the way I like to think about genomics is that it is reading the genome, and then gene editing would be writing the genome, right? And so this is what do we do with the right part of the read-write biological revolution that's kind of happening the first quarter of the 21st century. And so right now you can get a $200 whole genome sequence or whatever from a consumer company. It's not that difficult. So basically the read part, that is just a matter of capacity and will. We don't actually have the capacity to sequence every American very quickly right now because there's not enough, capacity in terms of from storage to also just like the machines that do the sequencing. But we could do it. We could brute force it. That wouldn't be an issue. The writing, though, we haven't done that much gene editing. And before we get to your piece, Antonio, which is like from August issue of MIT Tech Review, can you just give us like a general, like, I don't know, 100,000 foot sense of what's going on with CRISPR and the medical field? Because there have been like attempts, like some successful, to fix sickle cell. There's also like work on cystic fibrosis, although I think that, I'm not going to get into it, but one of the things is cystic fibrosis, and I know one of the reasons that a lot of people were not super focused on cholesterol targeting is the drugs are so good, right? So if you have a drug, why do the gene editing? But in any case, there's a boom. Yeah, go on.
Let me jump in and just answer that question. I mean, CRISPR is just really efficient. So to get you to the genetic modification of plants, plant seeds, mice, even some large animals had been happening, but it was all based on like an inefficient process, right? Called gene targeting. So basically you'd have a bunch of cells and you would sort of randomly mutate them or randomly try and add a gene, and it might work or it might not, but you'd have a million cells and you'd have some way to select them. So that's how you would get your change. That's not going to work in an embryo, right? An embryo is one cell. So CRISPR specifically, in terms of the germline editing, CRISPR is efficient enough that you could, for the first time, contemplate editing a human embryo, right? I'm going to fertilize the egg, and at that moment, I'm going to put in my gene editor, which, as you say, is this protein that cuts the DNA, and then targeting molecule that guides it to where the cut is. Suddenly, with CRISPR, it was actually possible to edit an embryo. And of course, it happened in mice and donkeys and, well, not donkeys, but monkeys, sheep, and so on. So it has just revolutionized genetic engineering, including of animals, made it a lot easier. In terms of where the therapeutics are, of course, like right away, and this is 2013, a bunch of companies were set up. There was Editas Medicine, Intellia, were two of the big ones, and CRISPR Therapeutics, probably the big three that were formed around that time. And they all set out to develop treatments using CRISPR. And so they had like some considerations. First of all, how do they get it into the body? You know, the sickle cell strategy is ex vivo. You're taking blood stem cells out of the body, you're editing them in the lab. Editas Medicine went for something in the eye. You could just like inject the CRISPR reagents into the eye and expect to get an edit. And then Intellia took a third path, and they use nanoparticles, just like in the COVID mRNA vaccine. They use nanoparticles and they wrap the CRISPR inside it. And they target liver diseases. And the reason they do is because the liver loves to suck up these nanoparticles. So they're sort of in the liver space. And then you need a target, right? You need a target. And what CRISPR is good at in this first generation, CRISPR 1.0, it's good at knocking out a gene. It's good at just like busting a gene. It makes what's called an indel. It just cuts the DNA. The DNA repairs itself, but with an error, which stops the gene from being transcribed. So it's just producing knockouts. So basically, this first generation of treatments is all about knocking out some gene. And how do you know that's going to have a function? Well, a lot of the times it's from population genetics, right? These big studies, surveys that you're talking about all the time, human population genetics, the GWAS, those are the types of techniques that can tell you where the cut should be made. So the beautiful example is Casgevy. It is this treatment from Vertex Pharmaceuticals. It's the first gene editing treatment approved in Europe, in the US, and it's for sickle cell disease. And the target that they have, it's kind of like a bank shot. They want to turn on fetal hemoglobin, which is like a different version of hemoglobin that you have. In utero, it gets turned off when before you turn 1. But they found you could turn it back on. And I think it's by sort of disabling the promoter of the repressor of fetal hemoglobin. So lots of like nice population genetics went into it. So they take the cells out, the blood stem cells, they edit them in the lab, and then they put it back. And these people are being cured of sickle cell. Like they don't have the symptoms, horrible symptoms anymore. But the treatment is also like an ordeal, right? It is an ordeal. There's a person already has died in a trial because it is a bone marrow transplant. So you're getting hit with chemotherapy. You're in the hospital for 2 months. I forget what the price is of Casgevy, something like 2.2 million, not including the hospital costs. So it's amazing, but it's really expensive, and it's yet to be seen how many people with sickle cell disease really want to go through this. So that's kind of where it stands with the drugs. And I would say there's a bit of crowding around the good targets. Like there must be 15 trials underway for sickle cell disease. And then there's a couple for liver diseases. But then the list gets really, really, and then there's a bunch that involve using CRISPR to make these cancer drugs involving T cells. But the list kind of tails off really quickly, which is interesting because there must be, people say there's 3,000 different sort of monogenetic diseases, rare diseases that are out there. And CRISPR really could be used for a lot of them, but it's not being used for them because it's just like the market isn't there.
Well, I mean, to be fair to the CRISPR companies, I always feel like, this is literally a 12-year-old field, and we're already talking about.
Yeah.
It's like a glass half full, half empty, because people are like, oh, the sickle cell stuff is so difficult and all this. But I'm like,
it's fast.
Literally, the beginning of 2012, you could not have imagined this. Like, right? It's not like there was a roadmap, right? And so now the fact that we're here, 12 years later, I think is incredible from the perspective of 2012, not 2024. Not for the people that are investing and the people that want cures as fast as possible, but cancer treatments, everything is improving over time. I think in another 10 years, price point's going to come down as they improve these techniques, and I'm assuming they're going to get better with fewer off-target effects, better delivery. In terms of what you're saying, that they're targeting the same things. They're looking for high penetrance stuff like big gene effects, right? With like crappy diseases. But as they get more efficient, it's cheaper, more precise, more accurate, because there's other Cases besides Cas9. There's still a lot of work to do in the field. I'm assuming that we will see that expansion beyond these core low-hanging fruit, right? I mean, this is like capitalism,
Yeah. Let me tell you about a couple of the one-off things that are going on, which are interesting and may direct your listeners a little bit to what's going to happen in the future. Like just recently, 2 different companies announced they're going to start a trial using CRISPR to target the hepatitis B virus. Like if you get hepatitis B, a lot of people just get over it, and it's kind of cured. Other people have sort of chronic problems with hepatitis B. It's like the top cause of liver cancer. Okay, this virus sets up in your liver. It forms like a little vesicle or something, and it just hides out in your liver cells. So it's hard to get rid of altogether. Even with drugs, the viral genomes are still in your liver, and they're still sort of producing some proteins that cause problems. So the solution possibly is a CRISPR drug, which is just going to target this virus, just like CRISPR used to do or does in bacteria, and they're going to aim it at the virus to chop up these genomes. And similarly for HIV, some people have tried it in HIV, see if you could give someone a CRISPR drug and wipe out HIV, which also, as you know, kind of hides out, and it's hard to get the last reservoirs of it out. So that's one area. The other one that leads into my favorite topic, like more eugenics, is a company called Verve, which has a CRISPR therapy that they're trying. It also goes into the liver, but what they're trying to do is knock out a gene called PCSK9, which is important in like cholesterol processing.
Yeah.
So population genetics, 15 years ago, researchers found some people that had like ultra low levels of bad cholesterol, just like super low. And they studied them, and they find out that they are all PCSK9 knockouts. They lack this gene, and it has this incredible effect, super low cholesterol. So that's what Verve is doing. And they're trying to treat people who have hypercholesterolemia. It's like horrible disease where your blood looks like butter. But if it works, they definitely, there's definitely this idea in the background is like, hey, everybody who's 20 years old should get this treatment, because then you have lifelong low cholesterol.
Yeah.
And the cardiologists are convinced like cholesterol is just like a poison that over the years through exposure, day after day, hour after hour for however many decades, it's going to kill you. If something else doesn't get you first, it's going to kill you. I mean, heart disease is the top killer. So right there, you have a potential intervention to eradicate heart disease. Now, is it that simple? Do you really want this treatment? Those things are undetermined. But, it's out there as an idea. And I just think it's interesting because if you say, I'm not only concerned about what I could do to treat disease, but I'm also going to open the door to enhancements or improvements. Well, then your menu gets bigger.
Yeah, I have to point out that a lot of times, as genetic variation, when you change a gene, when you change a functional part of the gene, let's put it that way. I just want to be precise here for the nerds out there. There are downstream consequences that you don't always see. We don't know what those could be. And so that will be a consideration. I mean, not just in genetics, like Ozempic, the GLP-1s, like Wegovy, Ozempic. These drugs are kind of miracle drugs in a lot of ways, but it's very early. We don't know what the long-term consequences are. Now, I do think with something like Ozempic, I think it's pretty clear at this point that it should be more widespread. It's literally turning around the obesity numbers in the United States over the last year. And we know what the downside effect of obesity is. Obesity is a disease, causes massive inflammatory responses. All these metabolic diseases are associated with it, et cetera, et cetera. So I think Ozempic's upside is just so clear that we probably should take the risk of what its side effects are going to be over the long term. A similar thing, obviously, with genetics, but the problem with genetics is, a gene edit, that's there. Whereas with Ozempic, you could just stop, with a drug, you could just stop in theory, and eventually it'll clear out of your system. But with a gene edit, that's there for you for the rest of your life. And so I think that's the kind of thing that people are a little worried about. So let's talk a little bit about your piece in terms of, this gene editing where we are now, his like attempt to come back, or his actually literal comeback, and kind of the way the scientists are reacting. But do you want to give like a little bit like a potted history, or just like tell people what he did in 2018 for those who need a refresher, and then talk about he was in prison for 3 years, now he's back, supposedly doing laboratories, supposedly has these projects. No one's sure what's going on, but a lot of scientists are pretty mad at him still. So just talk a little about that.
Yeah. I mean, similar to when I wrote about your sequencing of your older son, with the creation of the CRISPR babies, I mean, it was clear that it could be done. And so then my job as a journalist was like to take it from there. I'm like, okay, now I got to find who's doing it. And so that was a long process that took a few years. And eventually, in this fall of 2018, I did go to China to interview different scientists there who had been editing embryos, right? They had not been making the CRISPR babies, but they had been publishing reports, which were also scandalizing people, about editing embryos. And those reports were scary. I mean, the first time you see a scientific report with all this jargon in it, but the subject is like a human embryo, and you're changing the genes, I mean, it's really frightening to read. So I went to China to interview these different groups to, not necessarily to find the CRISPR baby, but to find what was the path that they were on. And it was pretty clear that, they were interested in this technology. They didn't have the same hangups as, the West did about it. And, they could get a Nature paper, right? They could potentially get a Nature paper. Like, it is ambitious. And so what the Chinese scientists were doing, where they were kind of filling in a vacuum. Like, the West had sort of said, whoa, this is too controversial. And some Chinese researchers said, well, that sounds like opportunity to me. So they were filling that space. And during that trip, I did encounter Jiang Kui He is how his name is said. I'm just going to call him JK. That's how he refers to himself. I did meet him, and he told me about a bunch of research that he was doing, preclinical research on, mice, monkeys, human embryos in the lab. He didn't let on that there was CRISPR babies. But what was happening was that very month, later that month, after I left China, in Hong Kong, there was going to be this huge second international genome editing summit, where all the important people in genome editing, Jennifer Doudna, and David Liu, and all these huge names, they all descended onto Hong Kong in order to debate principally whether or not society should move forward with germline editing, meaning editing of embryos, heritable embryo editing. So they were all gathered to ponder it and to debate it. But of course, He had already done it. And so a couple of days before the meeting, I just found his documents. Like, he had started to post some documents about it because he was going to try and get his paper published. And he had published an ethics statement, or rather posted an ethics statement. I should put ethics sort of in quote marks. The same ethics statement said, I'm going to win a Nobel Prize with this. He posted a sketch of the clinical plan, which was to knock out a gene called CCR5. And it's known that people who lack CCR5 cannot be infected with HIV. And actually, he had interesting reasons for doing this experiment. And then the last piece of data were some measurements on fetuses. It was cell-free DNA data. That is data that you collect to look at a fetus, to look at the genes of a fetus. So I knew right then that there was pregnancies, and it had some information.
Yeah.
7 months along. Once we published our story, then he quickly went on YouTube and he said, oh, I've actually made 2 children. There are 2 children. Forget Lulu and Nana, I think he called them. But his reasons for doing it are pretty interesting. HIV spread in China due to kind of like blood donation programs. They were using dirty needles. So different villages, like everybody got HIV. There's a huge stigma associated with it. And actually, people, men who have HIV, are not permitted to afford themselves of IVF services. So his proposal was like, listen, I'm going to solve your social problem. I'm solving your social problem because I'm going to allow you, first of all, I'm going to let you have IVF services through some trickery, deceiving people probably about who are the clients. So one thing, he's going to give them access to IVF. And the second thing is he's going to guarantee their kids cannot get HIV. So it was an interesting social proposal that he had. Of course, he did it illegally. Like there was different things that were done illegally. Like even on the draft paper, none of the there's nobody with an MD on the paper. I'm like, where's the obstetrician? Where's the IVF doctor? So it's still unclear. Like, did he, what level of deception was involved in making these children? It could be that the doctors just didn't know what was going on. And he was put in prison for that. The Chinese, of course, there's this huge uproar that he had gone ahead and done it before society's permission was in place. There was a huge uproar. He was roundly criticized by scientists, branded as Frankenstein, Chinese Frankenstein, and was put in prison for 3 years. The crime was basically, pretending to be a doctor, some version of that, right? Practicing medicine without a license.
Yeah, and like, what kind of? Do you know what kind of prison he was put in? I mean, was it like a pretty chill prison, or?
Part of the time he was in sort of a pretrial detention, and then he was in prison. And I have talked to him, and I asked him about the prison, and I asked him questions like, "Well, what were you thinking about in prison?" One of the things he was thinking about probably was how much he dislikes me because I caused an inconvenience for him by pushing his story into a global news cycle before he was ready. But, I'm very interested in what he does. I'm interested in him. And eventually, I guess he's willing to talk to me because, we're interested in the same stuff. He never answered really what happened in prison or what the prison was like. He's on a campaign to restore his name, to be recognized as a CRISPR pioneer, and on a campaign to bring his idea about germline editing back into the spotlight, to do it. You mentioned his Twitter profile. I mean, it's extremely interesting, almost like a work of art. He has all these pictures of himself just sort of standing in empty laboratories with all these slogans like heritable genome editing will save the world, or Casgevy is a failure, he says, referring to the sickle cell drug. I mean, it's kind of very interesting, but he's doggedly trying to make a comeback. And there's some people, supporting him, including me, because, I decided the article that we're talking about was for the 125th anniversary issue of Technology Review. And so the premise of this issue, that's how long our magazine's been around. The premise of the issue was, what can you say about what the world will be like 125 years into the future? I was like, man, I don't know. But changing evolution, intervening in evolution is going to be part of it. And so I decided to go back and kind of see where Dr. He was at, where JK was at. Because interestingly enough, he's the only one talking about it publicly. There's so much.
Yeah.
It's stigmatized. It's illegal. There's no funding. There's no pathway to make any money. So and it doesn't exactly work in embryos, which we can get into. But, for all those reasons, like nobody is talking about intervening in human evolution. But isn't that the point? Like we have evolved to the point where we can take over. Like, shouldn't we do it? Shouldn't that be the plan?
Yeah. Intelligent design.
That's right.
Well, I mean, so one thing you mentioned in your piece, you quoted the usual. I don't want to be dismissive here, but I would say that in academic science in the United States, I don't know. I mean, in the West, I'll say the West. There's a certain type of person now. There's not really. I would say really, it doesn't select for mavericks. It selects for people who operate within the bounds of the system. They do push the edges in the system, but they believe in the system. And you have a lot of people who denounced what JK did, and denounced this idea of gene editing, and whatnot, in like directed evolution, or they act like it's crazy, or like it's irresponsible. It has to pass the IRB board. But you said there that they also ultimately admit that this is going to be ubiquitous in a century or something.
Yeah, they do. Basically, yeah. That's where the story starts, is like the Promethean punishment of JK, right? day after day, he's tortured in the press and criticized. But when I went back to people, it turns out that the idea that he has, the basic idea, they also believe in, which is what? One, the embryo is easy to intervene in. It's just one cell, right? And then you're affecting the whole body. That is a starting point. Second of all, a lot of the people are talking to are human population geneticists, and so they definitely see the logic in surveying the entire human population, finding useful variants, and then those are the ones that you're going to install in an embryo. You don't need just the variation that you got. You could get the variation that other people got. Actually, JK, he has mentioned to me, it hasn't really been a focus of the news media, but this CCR5 mutation, the knockout, is common in Europe, relatively common. I think it's like 10%. And to be homozygous or two copies.
Yeah, my ex-wife has she's a heterozygote.
Okay. And then to be a homozygote, it's probably a tenth of that. So it's like, less than 1%. But, he did note, he's kind of enviously, he's like, wow, Europeans have this and the Chinese don't. But through technology, he can go and grab a useful variant from another geographic group and bring it into China. So, the people that I talk to, one of them is a kind of gene editor at University of California, Berkeley, Fyodor Urnov, his great turn of phrase. He's always appearing because he has colorful quotes, but he definitely thinks this is inevitable and going to happen, and including for sort of enhancements, because there's this long list of genes. People were tweeting about one just yesterday. They found some new gene that you only need, it's like a single base pair mutation, and you only need 4 hours of sleep a night.
Yeah.
People were just tweeting about it. They're like, yeah, we want this. So in the sort of Silicon Valley crowd, the people that were tweeting about it, that looks like a good idea. Like, let's all become Superman and get this variant. You don't have to sleep 4 hours a day. But the point is, this is existing genetic variation that exists in people, sometimes at a pretty high frequency. And because of the way population genetics works, you can actually see the experiment has been done. You can find someone of a particular genotype or find hundreds of them, and you can study their life history. So you kind of know the result.
The experiment, yeah. It's like natural. It's like natural experiment. We are, human population variation is, it's like a laboratory that's already there. The laboratory has been going on for millions of years. I think the one thing that people are concerned about, which it's not like a total concern, it's not like the biggest deal, is let's say that the short sleep variant is found in X number of people, and but it turns out that there's some sort of gene interaction where you don't know, but there's another like variant that's associated with that, where if you didn't have that variant, you get miscarried. Do you know what I'm saying? And so like it could be something where like someone puts this into them, and then all of a sudden they have all these like negative consequences because like their genetic background is different. You didn't know because there's already an experiment of the selection doing a miscarriage. So I think people are gonna test it on animal models or whatever first to make sure that it works, right? Probably put it in mice.
Of course. And in some of the experiments that are a run up to a regular CRISPR treatment on an adult, one of the first things you do is you make a germline animal with the change, right? Because you can intervene on the germline of an animal. So you make the mouse that lacks CCR5 or the monkey that lacks CCR5. Anyway, you wouldn't do that. Monkeys don't get HIV. But yes, germline editing is sort of one way to test in animals, is one way to test if these things work. But the other reason, let's talk about one gene that JK is talking about. And this is part of the reason that I interviewed him, because it's a good one. Back in 2012, Decode Genetics, which I'm sure you've discussed on your show many times, the gene hunting company in Iceland, they published a report. They studied the people of Iceland, and they found that there was a mutation or a variant in a gene called APP, and that the people who had that never got Alzheimer's, basically, and remained sharp into their old age. And in fact, there was some evidence that they even lived longer because they weren't dying of Alzheimer's.
Yeah.
So this is a single change in the genome, one letter of 6 billion, and it seems to protect against Alzheimer's. Why is that? I think people are studying it. This gene is in sort of this amyloid processing cascade, that's processing cholesterol and so on and making the amyloid plaques. And so, biology is just like a big Rube Goldberg machine. You know what I mean? It's just like a whole bunch of systems that have evolved together, and it's perfectly plausible that a small change just short circuits the development of these plaques or whatever the root cause of Alzheimer's is. So this is definitely demonstrated in humans. But to your point, when I called Carrie Stephenson, who's the CEO of Decode, quite a character, he was like, well, we haven't found that many people with this variant. That's one thing. The second thing is, to your point, it could depend on the background, the genetic background. Like maybe this variant is doing this in, people, in Icelanders, but is not going to have the same effect on a different background. And that is something that is hard to test. And so it sort of gets to the JK story. It's like, it's actually kind of impossible to test directly whether this is going to work. Partly with Alzheimer's, the experiment's going to take 80 years. And so that's sort of why JK just jumped in and did it, right? Like he's like, even when I asked him this summer, I'm like, well, how are you going to prove it's safe? He sort of just threw up his hands. He's like, there's no plan. Obviously, you can do animal models and stuff.
Well, I do have to say, I was talking about, this is a little bit of a tangent, but I think it's interesting. So I was talking to a friend of mine. Like today Republicans are pretty vaccine skeptic, I would say, leading. Donald Trump doesn't want to talk about Operation Warp Speed too much. But back when it was happening, I remember people saying, okay, so I don't know much about virology or immunology, but I had a friend who worked in that field, and he was basically saying, in February of 2020, during the early COVID pandemic, it's going to take 5 years to develop a vaccine. That's just, even accelerating it, Well, obviously it didn't take 5 years because, they dropped a lot of barriers that would slow it down, right? In fact, the mRNA vaccine, I believe, it was, I think they basically had it by the end of February of 2020. Then they went through some expedited processes. But if we had done challenge trials very early on, we could have had the vaccine way earlier than we did. And challenge trials would just be like, some people volunteer, right? They volunteer. And that's, some bioethicists, which is like, I think it's frankly a fake field, are like, that's like really unethical. But I'm just like, if like 100 people volunteer, imagine how many lives could have been saved if we had accelerated 6 to 9 months earlier the vaccine, right?
Well, let me just agree with you. Like I covered, there was an initial meeting in the White House where Trump was there saying, let's do it in 6 months. And, everybody kind of laughed at him. And even Anthony Fauci was in the meeting, like rolling his eyes. But the executives that were there, including a guy named Bancel from Moderna and the people from BioNTech and Regeneron, they were like, yeah, we think we can do it. And so in that case, Trump was right. Why did it go so fast? You didn't need the challenge trial. There was so much COVID going around that once they set up the trial, they immediately got their exposures, right? It is true that sometimes in a trial...
We had natural challenge trials.
Oh, yeah, exactly. That's why that was one of the key reasons when it went so fast. If people had not been getting COVID, it would have taken much longer, but everybody was getting COVID, so the trial went really fast. It was well done. It also speaks to, a technology set, which in the case of the mRNA vaccines had been kind of worked out. It included the nanoparticle, the knowledge that these mRNA constructs could be a vaccine, as well as some information about how to inoculate someone with a spike protein from a coronavirus that had been worked out. And it also, it's relevant to the CRISPR story and the gene editing story and even the germline story in a couple of ways. One is several billion people have already gotten their first, genetic modification drug, right? Okay, it's mRNA, it's temporary, but yeah, I mean, it's a gene transcript and everybody got it. So if there was going to be some kind of reluctance to like have your genes modified, well, we kind of got over it. Like people still can't wrap their head around it.
Yeah. We don't brand it that way. We don't brand it that way though.
In fact, if you said that on Twitter, they would block your account. But some people, like George Church, he agrees. Like, yeah, it's gene therapy. It is a form of gene therapy. But the technology set is similar to what is being used in the clinical trials for CRISPR drugs for adults. Like, it is the mRNA to produce the CRISPR protein packaged in a nanoparticle. It's the same thing. And so Fyodor Urnov made a good point to me. He's like, listen, right now, today, there's enough CRISPR for the whole planet. It's not like there's some kind of shortage of it, okay? It is very cheap to make. It is very cheap to produce. It's just mRNA. We've already given vaccines with an mRNA molecule in it, and they cost $25. So I asked myself, what is it going to take to, what is the cost of goods to like treat someone with a CRISPR drug, that's made in the same way, mRNA inside a nanoparticle? Well, the back of the napkin is, the dose is like 100 times as much as the vaccine. So, $250,000. It's cheap. So the table has been set. Like, there's no shortage of CRISPR. We've got these ideas from population genetics about what to do, and yet we're not really willing to take the step into the embryo yet. But I should say, one thing I learned was that a long time ago, a guy named Joshua Lederberg, very famous biologist, won a Nobel Prize later, he proposed something called euphenics. Like, instead of eugenics, he proposes euphenics, which is good phenotypes. So he's like, we shouldn't do eugenics because we're going to have to sort of live with the plans of past eugenicists. Like, what are we going to do? Wait around and see how it works out? So he proposes euphenics, and he gives the example like organ transplant. You have an unhealthy organ, well, you could get a good organ. And I think, what he was proposing has just become the biotech industry. You mentioned Ozempic. Like, what is that except euphenics, except good phenotype? So there is a trend in the CRISPR field for easier delivery. Like, right now, delivering it is kind of hard, but a lot of people are working on delivering it better and to the more interesting cells, right? Instead of removing your bone marrow and editing it in the lab, people are working on, little antibodies or stuff that could stick to it so it would get into your bone marrow and edit those cells where they live. So you can imagine a trajectory towards easy gene editing of adult bodies. Imagine it was just as easy as sending information on Wi-Fi. Then you could test it and see if it worked.
Yeah. You would find plenty of volunteers.
Yeah, right. Exactly. So this question of the germline editing in the embryos with all its problems, kind of my conclusion in the article is that it might be the issue just might be avoided if we develop, or if scientists develop really good ways of delivering CRISPR, because then, adults can just make a free choice and have whatever traits that they want.
Yeah.
I mean,
well, yeah, that's fair. I think that's right. I will say one thing with the germline editing. Obviously, there are people who, for ethical reasons, would never do selective abortion. But I think one of the issues preventing germline editing is just like genetic screening. Is a lot less. Well, I mean, very few scientists have a problem with selective abortion, right? So they think, like, why would you do germline editing when you could just select the embryo that has the wild type, or like, the non-pathogenic?
Yeah. Yeah. So people, right. In the National Academy report that came out a year before Hojenko did his experiment, they were looking for, what are the pathways or the reasons that we might do germline editing? And this came up. If you have, a deleterious mutation in your family, well, you're reproducing, you can go to the IVF labs, they make 4 embryos, 2 won't have it. Just choose those 2. And that happens. That happens. It's not widely used, but it happens. And so then they had to look for these like ultra rare cases. Okay. Like, both parents are homozygous for some disease. Both have sickle cell disease. So none of them have the correct gene. So these people literally cannot have a child that does not have the disease. They could justify the use of CRISPR in the first case. So what does that mean? Well, people have gone looking for those cases, right? And they exist. In a country as big as China, for instance, you can find deaf people who are married to other deaf people that have the exact same genetic lesion, right? So they would be in this situation. But it's really rare. It's pretty rare. In fact, somebody just posted, a guy, what's his name? He's one of the founders of Mammoth Biosciences. Maybe his name is Trevor. Anyway,
Wait, was it Ben Lamb or no?
Lucas Harrington.
Okay,
he's a sort of part of this UC Berkeley group. He's graduated from there, and he's now the head of a CRISPR company that does diagnostics. But for some reason, he was compelled to like put forth a big thread on Twitter about, boy, we really need regulation of germline CRISPR because, he thinks it's going to happen. Of course, I texted him. I'm like, well, what do you know? Do you know somebody doing it? He didn't get back to me. But, he also put forth this example of two people with sickle cell disease. And I said, listen, what I'm hearing from the fertility doctors is those cases are just like so rare. They're just so rare. Yes. Yeah. In a country like Nigeria, you calculate maybe at random mating there would be like 10,000 such couples or something, but it's not random. Like people kind of know, and they're like, they know to avoid this. So I just think that is just like a poor use of CRISPR. I mean, it'll never fly for that reason, just because the cases are too poor, or too rare.
Well, okay, so one way that I can imagine, okay, this is science fictional, but I mean, at some point, Elon Musk wants us to go to Mars. We will be a spacefaring species, and the issue with adults doing gene editing is a lot of the development is already done, so you can't change your bone structure and a lot of the other kind of like structural scaffolding of your body. I can imagine possibly someone who wants to change the color of their skin to, I don't know, purple or something weird, because skin cells are always turning over. Okay, like I can imagine that. But obviously, you're not going to get taller, because the bone is done. It's, when you're an adult, you're done. Unless you do a lot of crazy developmental things, which is that's a whole different game. But imagine you want to design the optimal human for space. Instead of having it evolve out, you could probably do selection for probably something like a Dinka build, like really long and narrow. There's I don't know, other sorts of things. I'm sure there's various things related to balance. With in terms of your inner ear and stuff, that there's variation probably. Who knows?
You'd want them to be small. I mean,
light.
A small space, and
okay, that's fair.
You leave some animals on an island for 3 or 4 generations. When you come back, they're a lot smaller.
Yeah. Okay, so small and slight, like maybe like pygmies.
Yeah, small, strong. Maybe you give them some extra copies of P53 because they're going to get hit by some rays out there in space. They get cancer protection. There could be some personality traits.
But we talked about this before we were recording, but they are. I mean, this is a very, very tough, tough nut to crack. But with the cognitive performance variation stuff, and CRISPR has to get way, way better. But in 100 years, I'm sure it'll be great. So let's just give that fiat, just concede that. You would want someone that's really good at visual spatial, just in space, because you're gonna be you're exploring a 3-dimensional space. You better be good at not getting turned around and stuff. We actually on the flat Earth, we have so few challenges compared to somebody that would be in a space station.
Well, not only that, but as you know, if you've read if you eat cereal in the morning, you read the cereal box and it says, here's the essential vitamins and minerals that this has. Well, why are they essential? It's because your body doesn't make them. You got to eat them.
Yeah,
but there's other organisms, I think they're called prototrophs, that all they need is some water and some carbon, and they make them. So people are in the laboratory redesigning human cells to make some of the essential amino acids that we don't currently. Now that would be good for the astronaut, right? Because you don't want to have to bring your whole space ring along with you with the cows and everybody walking around. You want to be able to survive off of sunlight and water. So you could think of extreme circumstances. I'm glad you brought up space, because in the original instance, George Church, this geneticist at Harvard, who's kind of into transhumanism, he was the one that circulated a list of these sort of superpower genes, the things that you would knock out. CCR5, PCSK9 we mentioned, another gene, you knock it out, you get extra hard bones. So he circulated this list of knockouts under the title rare genes relevant to an extraterrestrial environment. So he was actually initially coming at it like this is kind of like this list of space genes. And it's interesting because, I think that JK was influenced by that list. Like he chose, he looked at that list to see what the menu was.
Well, and so let's just like in terms of context and perspective, we've had a draft of a human genome for 24 years. CRISPR has been around for 12 years. As we're recording this, we are very, very early on. Like imagine, I don't know. This is like talking about like nuclear technology in the 1930s, where there's some basic science that's been established. There's some like precursors that have been done, but it hasn't all snapped together. I think that the full suite of technologies that we're talking about in terms of like widespread usage and the protocols and the standards and everything like that, that's like a couple of decades out. Like, what do you think?
Well, I did spend some time thinking, first of all, should we intervene in evolution? Like, is this an opportunity that we are destined for? And then if the answer is yes, the question is how to do it, right? The question is how to do it. And so in that same text that I told you about where Joshua Lederberg was talking about euphenics, the opening essay is by Julian Huxley. He's the brother of Aldous Huxley, who wrote Brave New World. And so he definitely thinks, that we are in the era of, self-conscious evolution. Like to him, it's like the biggest thing, one of the biggest things that ever happened. First the universe is formed and then molecules start to, replicate themselves, and then poof, here we are. So he was calling for now a global evolutionary policy, which could be social and psychological at one level, but also should be genetic. And so I was interested to see like what tools how was he proposing to do it? And so he was actually in this camp that, the geniuses should put their sperm into sperm bank bunkers. It's called germinal choice. And that women would then get pregnant with this sort of chosen sperm, which actually would work. I mean, it would work to propagate those genes, as you know, from like, in the dairy cattle, all dairy cattle are the progeny of like two bulls from the 1950s because they actually did this. Now, this is, I don't think, socially it's not going to work, for like chosen sperm donors to have all the children. But what are the other options of actually intervening? Like, what are the other methods? Your show, I have to say, is sponsored by Orchid, right? This is
it was. That's smart. Yeah.
This is a company that tests embryos and looks sort of broadly across their genomes for trait predictors, and then you could choose within your own genes, your own offspring, embryonic offspring, to see which one you like. So that's interesting. That's working. It's sort of crude in the sense that you only have like 4 or 5 embryos to pick from. On the other hand, you are picking all the genes at once, right? CRISPR, by contrast, is much more technological. You're coming in with precision and you're aiming for a big effect, right? With one gene, you're looking for a really big effect. I'm going to make someone who will never get Alzheimer's. So I'm also interested in sort of what are the methods that you're going to do about this. And I have to say, there was a pretty interesting tweet from Brian Armstrong. He's like one of these crypto people. I think he's the founder of Coinbase. And he is associated with a venture capitalist named Blake Byers, who's probably the descendant of Brooke Byers, a very famous person who was the B in Kleiner Perkins Caulfield Byers, which was one of the great venture capital firms. Anyway, Brian Armstrong is on Twitter. He's saying, hey, in Q1, I'm hosting a Frontier Bio dinner on assisted reproductive technology in the Bay Area. Bullet points: artificial wombs, embryo editing, in vitro gametogenesis, trisomy knockout, next-gen IVF. If you're a scientist or engineer interested in this field, apply to attend my Forbidden Technologies soiree. I added the last part. He just said, click here to apply. So here's a guy who is basically just throwing out a laundry list of like all emerging sort of taboo technologies at once, but I guess is looking for ways to bring them together into some kind of brave new world situation. Like, what are we talking about? We're talking about edited embryos, grown in artificial wombs, using germ cells like made in a kind of a factory. It's kind of interesting that this proposal is out there. Of course, the source is a little questionable. It's a crypto guy. He's like made money on Bitcoin or Ethereum.
He's like genuinely enthusiastic about this, and I know this because one of my closest friends from graduate school works for him at New Limit, which is his thing he co-founded with Byers, and is doing all this like life extension, anti-aging stuff. I know that a lot of people in the biological sciences kind of like roll their eyes at these like these crypto people, just these tech people in general. But I don't know. I mean, I guess what I would say is people need to chill out a little bit because, again, like the bureaucratic mindset, look at what happened with Alzheimer's research. Like the NIH wasted all this money on all this fake stuff. And so I think that there are opportunities and possibilities that people are missing. Just putting that out there. Most of this crazy stuff's not going to work out, but most crazy stuff doesn't work out in a lot of these tech fields. Like they have a different mentality. They're not trying to fund things that will get them the next NIH grant. When you do that, you have to do much more high probability things, which are usually lower yield, to be honest, right? And so I think someone like Armstrong, he's throwing out all these crazy ideas, but he's just like trying to see what sticks,
Yeah, I mean, yeah, right. I think his motive. He's got apparently a lot of money from the Bitcoin, so he's not restricted in terms of the financing. And I just think he finds it interesting. None of this stuff is like really a business, first of all. So it's not even a business proposal. It's really more of like a techno-social proposal. And I think it sort of fits under the umbrella of the stuff that Elon Musk has been tweeting about. There's a lot of tweets about the declining number of children, right? The fertility rate is going down. In Korea, it's negative or something. It's like really people are
0.68. Oh, actually, no. I think they got up to 0.74 maybe this year, estimates. So
right, 0.74. So the replacement rate, a couple or a woman needs to have two and a bit children. So people are way below it, and it just creates a demographic erosion of the demographic pyramid. So eventually it's going to collapse. You have all the old people and no young people. So is technology a solution to that? Not really, because, even if it's hard to get pregnant, it's also really easy to get pregnant. We don't need technology. It's not a technological problem. But I think, the thing that Brian Armstrong is talking about is sort of, it's assembling all the technologies in a way that could respond to Elon's demand for more people. And that actually gets back to Brave New World, because Brave New World, if you read it, the opening scene is in the London embryo hatchery, where there's a process to split the embryo. So they're making all these clones, and then some are the elite ones, and then other ones get a little chemical added to them to dumb them down, and those are the betas, and they wear black tunics, and they're the lower class. So, but if you take Elon seriously, then you're like, wow, if people are not having children, states might have to step in, right? Maybe the state should step in. And in which case, these technologies, embryos made in the laboratory and grown in artificial wombs without, parents, and that's kind of the way you would do it, right? If you just said, our society needs more people, so we're gonna manufacture them.
Well, I mean, there is so I actually, some of the podcasts I've done, when it comes to genetics, a lot of the stuff that's happened in the last 25 years is all of these different technological fields advancing simultaneously and kind of coming together in a synthesis, right? So, for example, concretely, DNA extraction, PCR amplification, you need a lot less DNA. You could get it from a lot of different things. And you can use whole genome sequencing that generates all this data, which could be handled by these, distributed computing and all these cloud services, right? So all of that rolled together creates this like suite of services in genomics that we can use today. Well, when you're talking about like artificial wombs, like these sorts of like complicated, gametogenesis from your own cells. All of this is going to possibly, I mean, it could come together and create kind of this new field of, the artificial production of humanity. And this is like obviously a very, it's a Brave New World, but actually, the most recent Superman movie explicitly, stated that this is how people on Krypton are made, and that Kal-El, and I forget what her Superman's mom's name is, but that they did it the old-fashioned way, which was like a total outrage. So Kal-El is genetically recombined and not planned, and General Zod was flipping his shit when he found out about that, because it's like blasphemy.
I was just cruising YouTube last week, and I ended up on some of the 1980s Superman movie, and General Zod is in there. Okay, to your point, a grouping of technologies. Well, I call it embryo tech, right? When it comes to the embryo, there's things that you could do to an embryo, like apply CRISPR to try and edit it, or apply a genetic test to the embryo to measure something. But the embryo at the same time also kicks off technology, right? Because something powerful is going on in there, right? There's a new life. Like two old people are making a young person. And so what are those technologies that have come out of the embryo? One is the whole idea of embryonic stem cells, right? You isolate the cell from the blastocyst stage embryo, and you have a cell that is immortal. It never dies. It keeps replicating. It is pluripotent. It can make every other cell, right? The stem cells that people are working with today in clinical trials are the same ones that were taken out of an embryo in Wisconsin in 1998. It's the same supply of cells. And then what else has come out of the embryo? Well, this whole technology of reprogramming that you said that Brian Armstrong and Blake Byers and your friend are working on at this company, New Limit. That's a new technology. Reprogramming, can you make cells young again? Well, that's of course what the egg does. That's what happens. And that's where that technology comes from. So think not only what you can do to the embryo, but this whole field of developmental biology as a gateway to technology that can address the supreme questions, not just reproduction, but immortality itself, right? I mean, I think maybe behind Brian Armstrong's tweet about wombs and embryo editing and stuff lurks this question of immortality, because that's a live question when it comes to the embryo, right? The human race is not dying out. We're the cells keep going.
Maybe the Koreans are, though.
Well, yeah, right.
No, if we have immortality, if we have immortality, I do think that inevitably does lead to controlled reproduction, right? Because if we have, let's say let's not say total immortality, but let's say we can live for 1,000 years, okay? And some people will die in accidents and things like that, but basically governments will be like, look, we can't produce too many people, right? So I think immortality inevitably leads to controlled reproduction.
It could, or it's the answer to falling reproduction. I mean, what is the reason why people are having less children? Well, it could be that they're living longer. The average length of life has gone up pretty quickly. Like a year.
You're basically saying the generation times could just extend. We would just rescale humanity so that it's 250 years would be a generation time as opposed to 25 years.
Yeah. I don't know. People might make different choices. Some of them might be more willing to jump on a Mars capsule, and take that journey. Or maybe they would become even more cautious, like, Ray Kurzweil waiting for the singularity, taking all his vitamins and stuff and trying to stay alive.
Dude, I've been knowing that he's doing that for 20 years. And he's waiting. Singularity is supposed to be here in 5 years.
He's still waiting. Actually, I used to work in Brazil as a reporter, and he came to Brazil, and I was all over him, asking him if he was wearing a bulletproof jacket, because in Brazil you gotta go around in bulletproof cars and stuff. So that was kind of funny. I went to see what his survival strategy was in Brazil. So yeah.
I mean, we definitely live in interesting times. So I guess the moral or the I feel like reading your piece, it's basically all of this stuff is going to happen, but people don't really want to talk about it. But the people in the know it's going to happen, but there's no plan set up because it's politically toxic. Even though, technology has a logic all of its own, right? and stuff can get snapped together and deployed in ways that you don't mean to.
Yeah. But, so techno enthusiasts from crypto space or somewhere else will descend on this area and they'll see an opportunity, just like JK did, to make some progress. Nobody else is doing it. On the other hand, let's return to the biologists for a second. In reporting the story, I did talk to the only 2 U.S. teams that have ever reported editing an embryo. And there actually are some problems. Like, it doesn't actually work the way you want it to. They've tried base editors, which is a new kind of editor, which turns out to be kind of toxic to the embryo. And in other cases, they're trying to make a simple edit, and instead, boom, they lose a whole piece of a chromosome. So, if you talk to the biologists, what's needed would be some funding for just basic research, like how does this work? And that relates to the second point that maybe a tech person might not appreciate, is that if you do something wrong to the embryo, you're going to get an abnormality, right? And that is really bad. Like, if you produce a baby that's sick and abnormal, like that's not okay. And actually, that's what did cloning in. Like 20 years ago, people were cloning sheep and cloning dogs, and people were saying, "Oh, we should clone a person, right? That would be cool." And why didn't that? And it's possible. You could do it, but why didn't it happen? Well, one reason was that it was demonstrated that when you're cloning cattle or something, a decent proportion of them are just abnormal. They have like a big, large size fetuses. Something's wrong with the placenta. Something's wrong with their lungs. So it would just be intolerable to do an experiment in which you would risk some kind of abnormality. I mean, like, who are you, Dr. Mengele? That's what people don't realize, is that, and that's why JK was sort of punished so much, is that he took that risk.
I do think, in terms of genetic engineering, I've always said one of the promises of genetic engineering is in animal and plant breeding, mostly because, candidly, we can tolerate a lot of faulty outcomes, before we figure out the good one. We can't do that with humans. Well, we could. That's the issue. We could. But, it's not ethical.
Well, people draw the example of IVF. I mean, yeah, it was tested in rabbits or something, but, they just did it in people. There was no way to, ensure its safety.
Yeah. Well, yeah, and this autism and other weird things. Although that might be ascertainment, right?
No, I don't. There's some cardiac issues. I mean, the signal, whatever the signal is, it's small, especially small compared to all these people that have been born and all the happy parents and all the lives that have been lived. The signal of health effects is like de minimis, I would say.
Yeah.
So, but still, yeah.
So you've been on this for not quite a decade, but you've been on this CRISPR beat on and off for a while. Do you think would you use a CRISPR technology? I mean, I don't know what your health issues are, but just for fun, I don't know. I don't know. Here's something. Give yourself your natural hair color back. You are of a certain age, like me. You have a lot of white hair. If there was a CRISPR technology where you just apply it to your scalp and your old hair color comes back, it somehow rejuvenates your cells, would you use it in 10 years?
Right. Okay. It's not so much hair color, because you can also get there. There's pretty good hair coloring systems now.
Yeah, but just young, luxuriant, silky hair.
I think what male pattern baldness, there's definitely CRISPR projects, gene editing projects, even reprogramming projects. Because if you could solve the male pattern baldness, which of course strikes in the prime of life, that is a huge market. Obviously, it doesn't work. Otherwise, everybody would be using it. In this case, I asked myself the question about the Alzheimer's change. Like, you know what? If I was having another kid and I went to fertility clinic and they said, we're going to make this one base pair change and the kid won't get Alzheimer's, having had some relatives with Alzheimer's, I would say, yeah, I would do it. I would do it. I mean, of course, I'd want to believe that it's safe and et cetera, but I would definitely sign up for that. That's good. That's like a vaccine against Alzheimer's. If that were proven to work, it seems like a no-brainer.
Yeah. As I was reading your piece, I was like, I want this. I don't, I don't have like particularly high risk in my family, but it doesn't really matter if you live long enough. There's just a high chance you're going to get it, right?
Right. Yeah, exactly. And people could learn their genotype for like the major risk genes for Alzheimer's. I don't think they do because, what can you do about it? But, it's definitely genetically controlled. So you might have a family background where, the odds are high, right? Because of other genes that you have. So yeah, I would do it. You mentioned hair, skin color is another one. The melanocytes or whatever that make skin pigment are not that far beneath the skin. Like there could be a crystal cream, you just rub them on, just bang, knock out those genes, you change your skin color. And so, there is a big market for skin lightening stuff. So if there's one that used CRISPR and work better, would people use it?
In Asia they would.
Yeah.
There's a lot of Asians. Like, hey, anybody who's got some funds who maybe we should like do this is like a VC funded idea,
there was I wrote an article once. This is sort of like a taboo topic that people don't talk about, but people did do it. They made cattle with a genetic engineering system inside the cattle crelox. But anyway, the point was that they could then do a brand on the cattle, and they could just write on the cattle. It would just change the fur color. So it's definitely within the realm of feasibility.
Yeah, this is. I think this discussion is going to be a lot less. I do believe in like 20. Was it 32? Wow. Sorry. Just like the 21st century is just racing by. I think it's going to be a very, very non-theoretical discussion. I think there will be stuff that has happened in the next 10 years that we will be able to talk about. And so we'll see. What else are you working on right now, Antonio? Like what's on your radar as we close out?
Well, I can actually read the list of things that I'm working on because Brian Armstrong tweeted it. Artificial wombs, embryo editing, in vitro gametogenesis, trisomy knockout has been on my list, next-gen IVF. So he actually just basically tweeted my story whip list. I'm interested in synthetic embryos, which is embryos made from stem cells, anything that is embryo tech. And I'm also working on a story about ancient DNA, which of course I've learned something from you about. Finally getting around to write a story about ancient DNA and see what I can learn about what evolution was up to in the past.
Nice. Well, that field, obviously initially it was just like one or two samples and individuals, but now you have genome-wide association caliber data sets, right? And so you could do genome. That's I think what you're like looking at in terms of looking at genome-wide association, selection. Again, this is like a field that you couldn't even imagine 25 years ago. Just like how far, and this is another situation where you have multiple different technologies that came together and kind of like synergistically amplified it by orders of magnitude what people could do. And I think this is relevant, obviously, you've alluded to it, for our discussion that we had today, because the ancient world had a lot of natural experiments, and a lot of things that we take for granted today occurred in the past and they're fixed. Like there's no variation, right? So
I always like to discover something new or at least new to me when I'm writing one of these stories. And so in this case, it's really the process of going, you're taking this time machine into the past and looking at past genes, and you can teleport them into the future or into the present, but then you can apply them, right? This question of genetic diversity is not just what exists today, but with ancient DNA taken from skeletons, sediments, cave floors, whatever, you are accessing all the genetic diversity of the past too. And that can be used and is being used in a genetic engineering experiments. Like, let's change some kind of crop. Let's use a gene that we find, when barley grew in Greenland, So that's actually happening. So some of this experimental evolution in the lab is using ancient genetic information, which is cool.
Well, I think with that, we were talking for a while. It was great catching up with you, Antonio. I hope people Check out your stuff at MIT Tech Review. In terms of CRISPR and all that, I mean, this is going to be all over the media. I hope people have a general sense of where we are right now in 2024, because the next decade is going to be a fun ride. I think it's on the whole for the good, but I don't. I've said some dismissive things about bioethicists because I think a lot of it's just hand wringing. On the other hand, we just need to have a broader public discussion of what's, you talked about trisomy. People are getting basically like Down syndrome fetuses are aborted at high rates, et cetera, and like there's this whole question like should we just like in Denmark and Iceland they just don't have any of them being born anymore. Is that good? Is that bad? Is that even a not even wrong question? These are things that are going to start to confront us. I do think, like for example, like if we like cured sickle cell and sickle cell disappeared, I don't think anybody would be sad about that, right? I mean, there's just certain things where like we cannot wait long enough to get rid of cystic fibrosis, sickle cell, all of these things.
Yeah. It's a public health menace, especially in places where it's common.
Yeah. So hopefully, like the science keeps proceeding in that direction because I think there's a unanimity, right? But anyhow, it was great talking to you. I will catch up with you online, Antonio.
Okay, Razib, thanks for having me on your podcast.
This podcast for kids. This is my favorite part.